Recording, and we're going to crank up the PowerPoint just here. And here we go. Okay, can you see this, Bruce?
Yep.
Okay. So what we have here is a little montage. This is Darwin here. And this is Michael Levin, who we're going to spend time talking about here. And Michael Levin's view of biology is this. And Darwin is connected to, this is the double helix of DNA here. It's intertwined and going up there. So Darwin's linked this way, and Michael Levin is quite knowledgeable on this, but he thinks this is a better view of biology. So we'll come back to this and you'll see why. So we talked about how physics had different paths that it took, and it didn't help that they took the wrong path. So, for example, after Maxwell had developed these equations, they said, well, this shows that there's propagation of electromagnetic energy, so there's got to be some medium for it, and they called it the ether. And they couldn't figure out how to measure it. So they ended up carrying out some experiments and interpreted the experiments to be that there wasn't any ether, which is in fact the wrong conclusion. The ether is really there, and if they would have kept with it, they would have advanced much more. And then when you get to 1975, they've got all these ideas about particles, and they refuse to go look to the east, to the ideas in the east for where they might come up with new ideas to fill out their ideas on the fundamental particles. So for 50 years, they've been stalled. So biology isn't any different than physics. There's a mainstream path to biology that's been followed, which we're going to run through. But then there's this alternative path, and that's the one that really doesn't get the press and doesn't get the funding and on and on. But in fact, this is probably the path that is really going to lead to some amazing things. So let's go back over to the mainstream path here. And what we have is what's called genetics or genes. And so these are entities that initially were unknown, but a monk in Austria carried out a bunch of experiments on breeding peas, and he concluded these things called genes did in fact exist, and his experiments suggested, how they would behave or interact with one another. And the genes is what Darwin tried to articulate. He said that you have evolution always occurs uh, as a survival of the fittest. So whatever genes that you have, whoever has the most fit genes, you're most likely to survive, and the other person doesn't, so they're going to die off. And the people left standing will be those with the best set of genes. So with that kind of as a backdrop, this was all in the 1850s, 1860s, 1870s. We fast forward to right after World War II. There's a lot of things that happened between then, but the net of it was that the biologists determined that the controlling mechanism for all of the cells was in something called deoxy nitronucleic acid, DNA, and recombinant nucleic acid. And these two things here controlled everything that led to these genes. The genes were actually a collection of proteins, just like the DNA and RNA are a collection of proteins. And so there was a big push to figure out what in the world is the structure of this? If these are controlling everything, what in the world's going on? So two people, Francis Crick and James Watson, who are very good theoreticians, worked with someone called Rosalind Franklin, who was a very good theoretician, but also an experimentalist. And she could carry out X-ray diffractions of these uh, proteins to come up with suggestions of how they would actually be put together. And if they didn't have her, they would have had nothing. Those pictures were the real tip-off as to what was really going on. So they came up with their ideas, and Maurice Wilkins was involved in terms of supporting or managing this group of people. So a Nobel Prize was awarded in '62 for this discovery, and guess who didn't get it? Rosalind Franklin. She had unfortunately died at that point, but she was just as important as Crick and Watson. And Wilkins, they give the Nobel Prize to at most three people. So that's what happened. Wilkins lucked out. So great. Now they have this idea of this controlling mechanism. They started to look at it and they realized that you have 50 trillion cells in your body, and every cell has uh, two strands of DNA. And when these cells split, when and for example, your skin every 90 days regenerates itself in your body. So all those cells are splitting all the time, generating new cells. So the DNA and what goes with it, the RNA, is actually coiled up inside each of these 50 trillion cells, and it's actually 6 feet long. Okay, 6 feet. So
Wow.
in the in when it when it
Just the
when it
average height of a
when it
male.
and it has 3 billion pairs. The DNA and is coupled to a mere strand. There's two strands of DNA. So you end up with 3 billion pairs of proteins there. And the thing that came out of this is that the DNA and the RNA have in it implicitly how this folds up. Because if you have 50 trillion cells, it folds up into something that's less than a millimeter long. So something that unwinds to 6 feet, then rolls back up into something that's under a millimeter. So there's something encoded here in these proteins that says this is how you do the folding, which is not obvious at all when you have 3 billion of these things. What's going on? So what happened was all of this theoretical work was done around 1950, and the rest of the time was spent really in, I'll just say, fleshing it out to get to the stage where you could do real engineering. And real engineering to me means that you could actually identify every protein out of the 3 billion proteins in one of these strands of human DNA. So that was called sequencing. What is the sequence of the 3 billion pairs of proteins? And two people led the charge on this. One, Craig Venter, set up a company, and his idea was, I'm going to figure out what the sequence is, and then I'm going to go patent it. And if you want to go do anything with those sequences, you have to come to me to be able to do anything. And so that led to a second group led by Eric Lander that was involved in this. And that was saying, well, this has got to be available to everybody. So they got all this government funding to go do this. And Venter was doing a lot of this on his own. But the net of it was sometime around, I want to say, they started in 1900, sometime around 2002, 2003, they declared victory. They said, we figured out most, quote unquote, most of this sequencing of the 3 billion proteins here, but it took them until, I believe, 2010 to actually flesh it all out. Um, they declared victory early, to put it differently. And uh, it took them a long time to carry this out if you're given some DNA, RNA, but this led to incredible engineering advances. So this genome sequencing activity required billions of dollars to go do this. But now you can actually get machines that will do it in under 10 minutes for under $100. And the cost keeps dropping. It'll probably be under $10, in 10 years. So it'll be trivial to go do this genome sequencing, which is what engineering is all about. What was funny to me was when all of this original work was done with DNA and RNA, there was a French biologist um, who looked at all this and he said, well, I guess there's nothing left to do. We've done it all. We've sorted out all the theoretical structures. And I said, yeah, all that's left to be done is to do the engineering so you can regenerate limbs. Or if you have a heart attack, regenerate heart muscle, or all these other things. But for the someone who's highly theoretical, that was like, how boring. That's really like engineering. Let the engineers go do that. So that's the mainstream path here. And this is where the bulk of the money goes. Here we saw this with COVID, that the US pumped a ton of money into this recombinant DNA as a vaccine to not go after the coronavirus. Now, that's all going on here. Darwin, Mendel, 1850, 1870s, 1880s. Okay. D'Arcy Wentworth Thompson, we saw uh, published a book in 1917, but he'd been thinking about it for decades. So kind of the same time frame. And he was looking at what Darwin had done, and he said, you're missing something here. There's all this structure where these forms are created and following these functions, and what you're doing with these genes doesn't begin to allow you to figure out how you get these shapes and so forth. So he started to really pose these questions, and they got picked up by Harold Burr, who was at Yale in the 1920s, 1930s. And Burr did something that Thomson didn't realize. He realized that there were electric fields. I didn't mention electric fields once in talking about this, but Burr saw that these electric fields were there and that they were in some way controlling how the DNA and RNA did things, or how for example, salamanders could regenerate a leg or a head or whatever when they had them cut off. There was some electrical mechanism there that no one was really familiar with. Crick and Watson were Brits. Sorry, Watson was an American, but they're there in England. Dennis Noble at the same time is there at Oxford, and he's picking up on these ideas, and he's saying there's real problems here with what Darwin is talking about. But this is getting so much money, it's going in this direction that it's like stopping a freight train. And Michael Levin, who's now at Tufts University, has been picking up on this. We'll spend a lot of time on what Michael Levin's done. It's truly amazing. And then to pick up on the bioelectric side, there's a Robert Becker who, after World War II, did a huge amount of work on this and all kinds of experiments trying to figure out how could you regenerate limbs and on and on. And the problem he faced was all the money's over here doing this. So he would write up a proposal when he's there at Syracuse University to go do some work in bioelectricity. And the other people there that would get these proposals would say, well, it's really interesting, but we're doing this sort of stuff over here. And it's all related to drugs and pharma, and it doesn't deal with bioelectricity, but it's interesting. So he just gave up. The the funding that he had just dried up because not because it wasn't worth pursuing, but because all the money went over here and the people were all caught up with this.
It's being just as dogmatic and political as religion, really. I was a medical and scientific proofreader for a few years in New York, and you could just see the bias and the camps that developed, and just as you're saying, where the money was going. And there were just trends. There were just absolute fashion statements that you had to stay within. Um,
Yeah, I'll give you a different anecdote. Um, years ago, I mean, 30 years ago, I was playing squash with a guy who's a very knowledgeable medical researcher and so forth. And I said, well, how many diseases are there that medicine has encountered? And he says, well, it's at least 25,000. Okay. And how many of those can we cure with all of this advances in pharma and so forth? And he said, probably 250. Well, if I look at 250 out of 25,000, that's 1%. So the other 99%, they're trying to, but they're not getting there. And that's why I think this other path is going to lead to things that'll go after the 99%, and this was why this will take off. But they don't want to hear that. Okay.
well, you can't patent it
Right.
very often.
So we're going to just do a quick review now of this, what I call mainstream biology. We touched on it already. So let's go into each of these people and see what's going on. So first we have Darwin here, who was a naturalist. He was a geologist. He had a theory of how rivers created all the erosion around him, not realizing that there were these plate tectonics, and also the fact that glaciers moved in and out to create a lot of the valleys and rivers that you see. So his ideas as a geologist got dropped, so he became a biologist. He went on this boat trip with the Beagle in 1837 and went all over the place, but notably to the Galapagos. And he felt here that all species of life have descended from a common ancestor. And this is really a fundamental concept in science, okay? And this comes about because all of the animals, and plants that we're aware of, all have DNA and RNA in them. That's how they function. So if everyone has that, there probably was some common starting point. And whatever else there was died out. It's gone. So he introduced a theory that this branching pattern of evolution uh, resulted from what he called natural selection. So there's a struggle for existence, and the things that are best suited to survive, they will survive, and the other ones that aren't will die out. And this leads to natural selection. And this also leads to selective breeding when you're looking to breed a plant for particular traits, or a dog, whatever it is. The same idea shows here. So he is felt to be one of the most influential figures in human history, and this is shown because the Brits buried him in Westminster Abbey. That's the penultimate in being a Brit,
What's the ultimate then?
being the king. So then we have this monk, Gregor Mendel, who lived in the middle of the 19th century. So he's there in his sanctuary, in his he's an Augustinian friar, and he's in this abbey in Brno in Moravia. And he was in a German-speaking family in that part of the Austro-Hungarian Empire. Today it's the Czech Republic. And many people view him as the real founder of the modern science of genetics. So what in the world did he do? Well, he saw these farmers had known for millennia that they could crossbreed animals and plants to get certain desirable traits. So he carried out a series of very controlled experiments, very well thought out, where he had peas and he did this in 1856 and 1863, and he had, it says here 7 characteristics: the height of the plant, the shape of the pod, the color of the pod, the shape of the seed, the color of the seed, the position of the flower, and the color of the flower. So one, two, three, four, five, six, seven traits. So he took the seed color as an example, and he showed that when you try to true breed yellow peas and take true breeding green peas and you crossbreed them, their offspring always produced yellow seeds. The green ones disappeared. But that was only in the first generation. In the next generation, the green peas reappeared at a ratio of 1 green to 3 yellow. So what in the world is going on here? So this led him, in explaining this, to come up with the notion of dominant and recessive genes. So in the example we just had, the yellow is dominant and the green is recessive, for that one particular example. Uh, and he published all this work in 1866, and he thereby demonstrating all of these invisible factors, which are today called genes, determine the traits of an organism. Now, the actual genes were only discovered in a long process that literally ended this year, when the last 3 of the 7 Mendel genes were identified in the P genome. All this work in sequencing of these genes, it's only this year that they were finally able to do all 7 of the genes of Mendel. So the genes here are collections of proteins, not a single protein, but a group of proteins. And of course, since he's doing this as a Austrian priest, monk, no one paid any attention to it. But at the turn of the 20th century, uh, what he was doing was rediscovered. The different people carried out experiments, and then they found Mendel's work and they tried to duplicate his experiments. They said, "My God, he did it all. We should have jumped on this." So Mendel is really the real deal. Now, we keep walking around these terms. So I'm going to talk about genetics and epigenetics. So genetics is the study of the genes, the collection of these proteins, and the study of their variation and heredity in organisms. So uh, this controls the evolution of these. Um, Mendel studied trait inheritance in these peas to see how traits were handed down from parents to offspring. And he observed that the pea plants inherit traits by way of discrete units of inheritance. And it's a somewhat ambiguous definition, but it's really what he meant as a gene. So the genes are intrinsic to the plant, the animal, whatever. So what is epigenetics? Well, remember, you have these plants and animals. They have a world around them, an environment, and so that's That's on top of the genetics. So the world around them can go change some of these genes, but that has nothing. It creates these variations that we're talking about just due to nature itself. So, for example, in 1945, the Nazis decided to simply starve everyone to death in the Netherlands, and they just gave them nothing but potatoes to eat for 5 months. And all of these babies who were born then were starved of all kinds of other nutrients, and that showed up in that whole generation of people. This is an epigenetic thing where the world around them changed. And so that a lot of them are lacking certain capabilities that the rest of us would have if they had had a good diet in the first place. But it shows up in many other ways too. It usually involves a change that isn't erased by any sort of cell division, and it affects the expression of the gene. A lot of people thought that the genes were it, and nature couldn't do anything. And in fact, there was a guy that Stalin had who uh, who said, no, it's the other way around. Nature influences everything, and the genes are influenced by this. But a lot of people, I think Stalin decided to kill this guy, uh, which is one of the problems of being too close to Stalin. But anyway, you can see there's nature and there's nurture. Nature is the genetics and nurture is the epigenetics. So to summarize, we've all got these 50 trillion cells in our body, and inside each cell, there's this so-called chromosome, which is here shown as these two strands of proteins that are in humans 6 feet long, and they're when the cell divides, this thing splits in two and then gets replicated. So each of these strands gets replicated to have another mirror strand, and they go into each of the two new cells that comes out of this. So uh, this was really in doubt until right after World War II, when Watson and Crick were doing this work. They wondered how in the world can cells divide, and then they discovered this protein here, and they said, voila, this explains everything. So Crick, who is a real dominant figure in modern biology,
so to speak.
What?
So to speak.
So to speak, yes. Um, he was a physicist and actually uh, met Watson. They were there. I think they were there at Oxford. I think, um, and so he ended up not only getting this Nobel Prize, but he influenced a lot of the research directions in modern biology after this for decades. Uh, James Watson is, I think now he's at whatever it is. Uh, uh, I want to say Cold Harbor Springs, but he also has some ties to San Diego. Anyway, he's the other one that got the Nobel Prize. But the thing is that Watson wrote a book where he tried to describe how in the world he and Crick had come up with this idea for this double helix. And the book is one of these books where it talks about everything. So you get the idea that really Watson went over to England as part of a postdoc, and he's going to the pub every day and having a few pints of bitter, and and Crick is there. Crick is a good Brit, so he's at the pub, but the two of them really are just theoreticians. They're just kind of screwing around. And it's really the next person here, Rosalind Franklin, is the one who did the X-ray crystallography work here, taking these X-rays and of all kinds of things, not just DNA and RNA. Here we go, deoxyribonucleic acid and ribonucleic acid. Okay, there you go. And then other things like viruses, which are pieces of proteins or coal or graphite that have a lot of structure to them. And so everyone appreciated her work on coal and viruses, but she never got the credit that Watson and Crick looked at all of her X-rays of various crystals of DNA and RNA, and that suggested to them what the right structure was. So, as always, the men looked out for the men, and the woman came out on the short end. And then Wilkins uh, ended up, and he was the one who had Rosalind Franklin doing these X-ray studies, and he also uh, was engaged with Watson and Crick because they were all over this trying to figure out what was going on. So there you go. Let's now go forward to the Human Genome Project. This is where I think the biologists were always in envy of the physicists. The physicists had this Manhattan Project, and they built this atomic bomb, but there was all kinds of things that came out of that, in particular doing particle accelerators to look at what makes up the pieces of what's inside the atom and the nucleus of the atom. And so the physicists were always building bigger and better accelerators and grabbing all this money based on the original project, which was the atomic bomb. So the biologist said, well, we can do the same thing. We're going to put together the Human Genome Project, okay? And we're going to sit there and start in 1990. And this says it was completed in 2003. But it was a huge collaboration of not just the US government and all kinds of uh, universities in the US. There were many, many countries that were involved. And they declared victory on April 14th of 2003 when they felt that they had sequenced or they had an idea of 92% of the genome. And the complete genome was achieved in May of 2021. And there's only 0.3%, but 0.3% of 3 billion. Okay, so uh, if I take 3 billion, 1% of that is 30 million, and a third of that is 10 million, 10 million base pairs of proteins are still unknown. Okay, so these numbers are staggering that they can even begin to do this. So what happened here? The idea was to have a complete set of all the nucleotides, all the proteins that were there in this strand of DNA, and the genome of any given individual is unique. So, then the question is, well, if it's unique for any given individual, like that's why we're all different from one another, then what in the world is going on here? Well, this should show you what things are there in this strand that make you unique, that make you you, and you not look like everybody else. So sampling this thing was done from a small number of individuals. For example, Craig Venter, who we talked about, and we'll come to in a second, he actually had his DNA sequenced here. He was one of them, and so forth. And they discovered that there's 23 human chromosome pairs. So there's 22 pairs, and then there's the sex chromosomes, which are in a different category to make you male and female. So the finished human genome is really a mosaic that doesn't represent any one individual, but the utility comes about from the vast majority of the human genome is the same in all humans, and so forth. So this was a huge activity. Now, with the money that's thrown into it, you can again buy relatively inexpensive machines and do this whole sequencing thing in, like I said, 10 minutes. And it's just going to drop to nothing. So here is Craig Venter, and he led one team of trying to sequence the human genome, but he did this with his a private company that he founded called Celera Genomics. The whole idea was, again, to sequence the genome and then patent different pieces, different genes, so that if anyone tried to do anything, they'd have to come to you to get a license. And this was constructive. I mean, it drove a lot of the people on the academic side to try and say, we got to beat this guy. He's the real deal, and he's very smart and capable, and so forth. So who went up against him? Well, it was Eric Lander is the one that I'm familiar with, who is at MIT, and he received one of these MacArthur fellowships. MacArthur Fellowship is given to a select group of people, I think maybe 40 people every year, and they're viewed as geniuses. And this gives them funding for 5 years to do whatever they want to do. So Lander founded a whole research institute in genomics, and he was the one that led the government-funded activity on the Human Genome Project and so forth. What I know Lander for is the following, that MIT has a core undergraduate curriculum. When you go there as a student, you have to take 4 semesters of phys ed, 4 semesters of humanities, 4 semesters of physics, 4 semesters of mathematics, typically calculus and differential equations, and then 2 semesters of chemistry. So the only thing that's open to you in your first 2 years is to take one elective in your sophomore year and in the fall and the spring term. That's it. It's cast in stone. And Lander showed up and he said, "Well, with all of this activity in biology, how could you say that you're graduating from MIT and never having taken a class in biology?" So he went and he got the chemistry people to give up one of their 2 classes in the core curriculum. So now you take 1 semester of chemistry and 1 semester of biology. And he created the introductory biology class and so forth. He also was the science advisor to Obama and to Biden. So let's look at the alternatives now. This is, I think, the interesting part. And Marjorie did a great job setting the stage talking about D'Arcy Wentworth Thompson. So we're going to have Marjorie pick up, and we'll just do a little refresher on D'Arcy Wentworth Thompson.
Okay, just to backtrack a little. He was born May 2nd, 1860, and lived to June 21st, 1948. So he was overlapping with Darwin, but younger. He was his junior. He was a Scottish biologist, mathematician, and classic scholar. He was a pioneer of mathematical and theoretical biology, traveled on expeditions to the Bering Strait, and held a position of professor of natural history at University College in Dundee, Scotland, for 32 years, then at St. Andrews for 31 years. He was elected a Fellow of the Royal Society, was knighted, and received the Darwin Medal. I wonder how he felt about that, and the Daniel Gerard Elliott Medal. He's remembered as the author of the 1917 book on growth and form, which is about this thick. You can't see me.
It's
Inches thick.
3 inches thick, at least.
On growth and form, and it's just right off with illustrations, wonderful illustrations, which led the way for the scientific explanation of morphogenesis, the process by which patterns and body structures are formed in plants and animals. And he saw it in such mathematically precise terms. I think that's what really set him apart. Thompson's description of the mathematical beauty of nature and the mathematical basis of the forms of animals and plants stimulated thinkers as diverse as Julian Huxley, C.H. Waddington, Alan Turing, Rene Thom,
Claude
Claude
Lévi-Strauss.
Lévi-Strauss, Eduardo
It was
Calzoli,
Le Corbusier.
Corbusier, Christopher Alexander, and Mies van der Rohe.
So let's just, this is a link to a video. I don't know if this is going to work, so let me see. I'm just going to click on this. Now, the question for Bruce is, hold on, let me go. I don't trust this. Let me go get here it is here. Yeah, we needed to share sound. That's what I was missing. Okay. So now let's see, where did the sound go? It's here.
Like my personality when I'm on stimulants.
And this is a little short video that reinforces what D'Arcy Thompson really did.
A century ago, in 1917, famous British mathematician biologist D'Arcy Thompson published a book called On Growth and Form, where he presented a number of interesting ideas which are still quite relevant to us today. One of them is the theory of transformations, where he applied mathematical models and mathematical thinking to connect biological shapes of living organisms to each other. When he took biological shapes of existing species of crabs and leaves and other animals and plants, and he showed that by geometrically transforming them, he could connect them in logical ways, which was very interesting because he showed that the shapes, biological shapes, are not random. They are connected by mathematical logic, which to him meant underlying physical and genetic principles that generated them. There's a very simple mathematical principle that he applied. In fact, he borrowed this principle from a German artist and scientist from a century before called Durell, where he took, who used drawings, and he used point-for-point transformations from one human head drawing to another drawing to show the relationship. And one of the most famous examples, D'Arcy Thompson, he used a head shape of the early horse ancestor, Hipparion, and he transformed it via shapes of intermediate species into the shape of a skull of a modern horse. And this is one of the most powerful examples of a transformation that essentially betrays evolution, from a primitive form, from a primitive shape, onto the modern form, onto the modern species. We're still using theory of transformation today. We call it geometric morphometrics, where we use landmarks and sometimes landmarkless approaches. And the main goal here is to understand how the biological shapes are related to each other. In our own work, we use this approach to understand how shapes of beaks in Darwin finches, for example, evolve over time, where we applied so-called F1 transformations, the very transformations that D'Arcy Thompson used, to see if they can be used to explain evolution of Darwin finches' beak shapes. F1 transformations are a hierarchical set of transformations which are used in geometry to connect geometric shapes. The most simple of them is called scaling, where we take a square grid, a shape on a square grid, and you stretch it or squeeze it to transform that shape into another shape. The next one, which is a bit more complex, is called shear, when you take a square grid and you make it oblique. And we showed that these two transformations, scaling and shear, they could explain all the variation of beak shapes in all Darwin finches. In fact, they explain variation of beak shapes across many of the songbirds, where the more common changes are based on scaling, where the beak becomes longer, becomes deeper, becomes wider. And the shear-based variations, when the beak curvature is changing, are much more rare, and they tend to be running more in families or in genera as opposed to species. We found that these two F1 transformations, scaling and shear, again, the very transformations that D'Arcy Thompson used, could successfully explain the entire variation of very highly adapted beak shapes in Darwin finches. During the time of Thompson, which was in early 20th century, it was quite this idea that you can connect biological shapes to each other using geometry, using mathematics, was very new and very striking. And it took some time for the scientists to realize that this ideology could be applied more broadly than just to a few chosen examples. Through most of the 20th century, this new field of what we call now geometric mathematics was very slowly developing, with more and more interesting and useful methods introduced by scientists. For example, one of the key innovations was using landmarks placed on what we call homologous points, that is, the same points on the animal or plants, which are evolutionarily related. If you place landmarks on them, then you can connect these landmarks into what we call a wireframe structure, and then see how this outline of a biological object changes over time. Another breakthrough was that we can use theory of transformations to understand how biological shape changes during development, individual development, from an embryo to all the way to adult through juvenile stages. And the fact that one can seamlessly connect biological shapes through development, and this will often be informative also to think about what happened during evolution, is a very powerful approach by itself. So it's in more recent times, in the last 20, 30 years, geometric morphometrics, when we used both evolutionary source of data but also developmental from the same species, is giving us new insights into how evolution proceeded or how to interpret developmental data more accurately. Geometric morphometrics, again, the field which was established by D'Arcy Thompson, is an incredibly important part of science today. We use geometric morphometrics to understand how biological shape changes, for example, from normal to abnormal in medicine. So when we look at the shapes of skulls and other bones in humans in normal conditions, but also in some of the abnormal conditions, that can be a very powerful way to understand what happened during disease. Again, I mentioned evolutionary applications where we can compare biological shapes using genetic transformations using the same, what we call within the same morphospace. Again, the concept which was invented by D'Arcy Thompson. Genetic morphospace, morphospace is the mathematical space within which biological shapes of different organisms can be compared meaningfully because we use the same landmarks, and we can understand how particular parts of this animal or the entire animal shape changes within the same geometric space. So we can use this morphospace to understand evolutionary changes. We can use the same morphospace to understand changes in development. In fact, we can combine developmental and evolutionary shapes together to understand how these two processes are related to each other. In our own work, we were able to use geometric mathematics with landmarks placed on skulls of multiple species of archosaurs. These are animals related to birds and crocodilians, to understand how their skull shapes changed during evolutionary time. And by adding skull shapes from juveniles and embryos, we're able to show that the origin of birds from reptiles, from these non-avian dinosaurs, which look more like reptiles, can be best explained by a process called metamorphism, where the descendants have the juvenile shapes of their ancestors. That is, the first birds, such as Archaeopteryx, the skull shape, again, geometrically defined skull shape of first birds, is very similar to the skull shapes of the juvenile non-avian dinosaur ancestors. That is, the very late embryos or very early hatchlings of the non-avian dinosaur ancestors of birds. And this was all done within the same morphospace. We used the same landmarks. We placed them on the same positions within the skulls of the species representing the entire lineage of archosaurs, from early archosaurs through dinosaurs all the way to modern birds. And when we combined all this data in the computer within the same morphospace, it was very clear that modern birds and first birds, they were in what we call the baby space. That is the part of the morphospace which is occupied by shapes of embryos and juveniles of the non-avian dinosaurs, which had much more reptilian appearance. Geometric morphometrics made a huge amount of progress in the last 100 years since Thompson. For example, we're very active in moving from two-dimensional morphometry, where we look at the outline of the skull in 2D, or outline of the typical bone, or for example, leaf shape in 2D, towards more three-dimensional, or in four-dimensional morphometry, where we look at the computer tomography scans. We use X-rays to generate high-definition three-dimensional images of, in our work, for example, of skulls of vertebrates, such as reptiles or mammals, and we place landmarks both externally on various external landmarks, but also internally. That way we can understand how the shape of this biological object changes in its full complexity in 3 dimensions. We can also look at the evolution and development of internal structures. For example, we can look at the changes in the developing brain. We can look at the changes in the developing olfactory structures, the eyes, and see how these different internal and external parts are integrated and how they change during evolution and development together.
Okay, so there you get a little touch of what's going on. Thompson didn't have the computer graphics we have today, and so you can do these transformations that he was talking about of stretching or torquing, or you can just translate something, put something side by side, and then overlay it on top of the other thing. You can see how much they line up. Okay, that's the power of having these computers available to you. So that what Thompson did was amazing, paper and pencil, but we can do much better today, and that's what you'll see as we go through the rest of the stuff.
We're still adding depth to his ideas 100 years later.
Absolutely. So, okay, can you see this, Bruce?
Yep.
So I have another one here on Thompson. Let's see if these things are really, great videos. It's amazing what's out there. Let's see.
We're finding so much validation.
Where did it go? Here it is. Oh, wait, I forgot to check one thing.
And published her book in less than 30 days. I never thought it could be this easy. I heard about Spine's book publishing platform from a friend who actually published 3 books with them, and I was super impressed after seeing the final results. After what she told me about how Spine's publishing process went behind the scenes, I wanted to check it out for myself and show
Here we
you
go.
how it looked.
Now this one we just did. Hold on a second. Going to close this.
But he was light years ahead of Darwin in so many ways and so marginalized. You know, that's what I got when I researched him. It was sad. It was really sad to see how his whole line of thought, which I thought was so much more creative and spiritual, really in its own way, that it was just dismissed.
No, wait, I just did this. Why is this? I must have screwed up. Hold on one sec. Hold on a second. The wonders of here we go.
I feel like for me, it just opened up this, it was like this pow, this
Here it is.
different view of
Challenger specimens for his
the,
museum.
of the divine, of what's going on in the
Hello, I'm
universe.
Matthew Jarron, Museum Curator at the University of Dundee, and welcome to the latest in a series of short films in which I talk about some of the highlights of the University Museum collections. And today we're going to be returning to the D'Arcy Thompson Zoology Museum to look at some of the smallest things that we have in the collection, specimens of minute single-celled organisms which fascinated D'Arcy throughout his life, called foraminifera. So they're known as forams for short, and they're microscopic creatures that live mostly at the bottom of the ocean in the sediment on the seabed. Indeed, their shells make up quite a significant part of that sediment. What that means, of course, is that they often wash up on beaches, and so if you're a keen naturalist like D'Arcy, you can try and find them and study them. D'Arcy's love of natural history began in childhood. Here he is at the age of 5 in a painting by his uncle, George Clark Stanton, which is in the University of St Andrews Museum collections. With some of his school friends in Edinburgh, D'Arcy formed a group called the Eureka Club, and they spent their Saturdays exploring the local area for plants and fossils. Beachcombing was a common part of their activities. As he grew older, D'Arcy joined the Edinburgh Naturalists Field Club and the Edinburgh Geological Society, and one of the first talks that he gave to them was about forams. D'Arcy went on to study medicine at Edinburgh University, but quickly realised that biology was his great passion, and after 2 years he transferred to Trinity College, Cambridge to study natural science.
Start the new year with big savings. It's the perfect time to focus on your health, and we are making it easier and more delicious.
What really fired his enthusiasm at Edinburgh was learning about marine biology under the legendary Charles Wyville Thompson, a veteran of several marine expeditions, including the deep-sea explorations of the famous HMS Challenger, and indeed D'Arcy later acquired several Challenger specimens for his museum in Dundee. We know from D'Arcy's surviving notes that Wyville Thompson's lectures on marine invertebrates included the study of foraminifera. D'Arcy took up the chair of biology at University College Dundee in 1885 and quickly began to build up an extensive museum collection which he used both for teaching and for research. He generally spent his summers staying with family in Ireland, and it was from there in 1889 that he wrote to one of his students, Mary Lily Walker, telling her, "I have taken to mathematics and believe I have discovered some unsuspected wonders in regard to the spirals of the foraminifera." And this is really important because this is the earliest reference we have to D'Arcy's interest in mathematical biology, which would eventually lead to his landmark book on growth and form, published in 1917. D'Arcy became increasingly convinced that the laws of mathematics and physics could be used to explain the growth and the shape of living organisms. At the time, this was a radical concept and certainly not accepted by the mainstream of biology. So what was it about forams that gave D'Arcy these ideas? These are some of the slides that D'Arcy had of foram specimens, and if we look at one of them through a microscope, this is what we see. Each foram grows a shell around it called a test, usually formed by secreting calcium carbonate onto their surface, and these tests grow into amazingly complex shapes. D'Arcy acquired hundreds of foram specimens to study, though only a fraction survive in the museum today. Many are undated, but a few come from the Challenger expeditions of the 1870s. After that, the earliest examples are probably these ones from Canada, prepared in 1895-6 by the wonderfully named Oregon Columbus Hastings. Later examples include this carefully prepared slide of 16 separate species by the London firm W. Watson and Sons. Of course, minute organisms like these aren't great for displaying in a museum, so D'Arcy also acquired a set of models made by Václav Frick of Prague. Frick was a natural history dealer who specialised in carefully prepared fluid specimens such as these ones, but who also made models of microorganisms. D'Arcy's collection of forams really took off in 1894 when he began to correspond with the oceanographer and microscopist Arthur Erland, at that time regarded as a leading expert on forams. In 1906, D'Arcy acquired a research ship, the Gold Seeker, as a result of his role as scientific advisor to the Scottish Fishery Board and head of the Scottish arm of the International Committee for Investigation of the North Sea. Erland was one of various researchers invited to join him on expeditions around the Scottish and Irish coasts. These are some of the slides we have prepared by Erland from specimens collected on the Gold Seeker. Erland discovered various new species on these expeditions, one of which in 1909 he named Technitella thomsoni in honour of D'Arcy. Erland published much of his research in collaboration with another foram enthusiast, Edward Heron Allen. But although D'Arcy was good friends with both of them, he fundamentally disagreed with their ideas. Like many naturalists at the time, Erland was puzzled by the variety and geometric precision of the shapes and patterns of foram tests, writing, "So far as I can see, they could not have been formed under any evolutionary scheme, for they can hardly be of any service to the animals." Well, D'Arcy also believed that this had nothing to do with Darwinian evolution, but he couldn't accept Erland's conclusion, which was that this could only imply the existence of selective or discriminative powers, a kind of intelligence. Well, as far as D'Arcy was concerned, this was preposterous. Writing to the biologist Edward Stuart Russell, he complained, "What I dislike most of all and lose all patience over is the attempt on the part of men like Arthur Erland to import the notion of behaviour, of taste, of skill, of incipient art into such things as the building of a little foraminiferous agglutinated house, before they've even given the least attention or thought to the physical processes involved and the precise inorganic parallels." He was even more brutal about Edward Heron Allen. Writing to the mathematician and physicist Joseph Larmor about the poor standard of papers being published by the Royal Society, he picks on this article by Heron Allen from 1915 as an example. "Here we have the old story that the little particle of protoplasm displays an astonishing intelligence in collecting and arranging particular kinds of sand grains, sponge spicules, etc., wherewith to make its shell. The whole argument is a piece of the grossest old-fashioned ignorance. The few biologists who know a little physics have known for years that the whole thing is a surface tension phenomenon. It's extremely annoying to see such rubbish printed here." In his book on growth and form, D'Arcy examines various types of foram in great detail and makes numerous parallels with inorganic forms such as water drops, bubbles and salt crystals, showing how simple physical processes can explain these complex geometric forms. In particular, he was fascinated by the logarithmic spiral patterns of certain kinds of foram, such as Globigerina, and here are images taken from some more of D'Arcy's microscope slides of that genus, so these are the very specimens he would have been studying. D'Arcy is now recognised as a key figure in foram studies, and forams are still actively researched today. Since they first evolved, between around 900 and 650 million years ago, the tests of dying forams have continuously fallen on the seabed in vast numbers, where they then get preserved in fossil layers. This has created an unparalleled fossil record, which enables scientists to understand the evolution of other species found alongside them. When the oil boom kicked off in the 1960s, it was studying forams and other microfossils that allowed the discovery of potential hydrocarbon deposits. Today, forams are used as a key indicator of climate change. As well as the slides and models of forams, we also have a set of paintings by the artist Roger Wilson that were inspired by them. And in the link entrance between the Carnelli building and the old medical school, you can see Peter Randall Page's beautiful sculpture called In the Beginning. Inspired by cells multiplying, it's made of Kilkenny limestone and contains numerous fossils of forams and other microorganisms. The Irish coast is still one of the best places to find forams today, which of course takes us back to where we started with D'Arcy studying the forams that he found there in the summer of 1889, without which he might never have been inspired to pursue his interest in applying mathematics to biology, and his book on growth and form might never have been written. Well, that's all I have time for today. Hope you found that interesting, and join me for another story coming soon. Bye for now.
So there you go. Just try to give you a little different taste of D'Arcy Thompson. Truly an amazing individual. Now let's go back to, let's see, is this it?
Well, isn't that just, to my mind, just a lovely contemplation of creation? And that it is precise, it is mathematical, but it's also beautiful. I mean, he didn't miss the beauty at all. No, why are they, at all considered mutually exclusive? And why, I don't know, I want to say, why shouldn't science be precise and beautiful?
Yeah, and then he's also arguing, how in the world can you have these things evolve? Like what is the mechanism? For example, I went to the Galapagos, a long time ago, 77 or whatever, and what Darwin did when he was there, there are these birds called boobies, and some of the boobies just go foraging by the beaches out maybe a mile or two, okay? And those boobies were noted to have blue feet. So then they went looking in the water and they discovered the boobies were going after these little creatures in the water that were blue. And so over a period of time, their feet became blue, and it was this virtuous cycle. They would always be looking for these creatures who would dye their feet blue. Then the next type of booby had red feet. And they were boobies that went like a mile or 2 miles to 4 miles, 5 miles out from the land. And they were eating these other creatures. And so their feet, so, you're looking at this and you're going, what does this have to do with evolution? But it was an example of this epigenetic phenomenon that the nature influenced the genome to take the feet and grab onto all this stuff. And then I think there was a green-footed booby that went further beyond all of them. So, and Darwin saw all this, and he noticed like the beaks of the boobies varied because it was the creatures they were going after were softer. I think what closer in and a little harder further out, so you needed a stronger beak. So the beak got stronger, and so it goes. So let's now go to this.
But what I love for myself was the is contemplation that that can be the divine at work. You don't have to be talking about a man with a long beard sitting on a throne and measuring it all with a slide rule and a protractor and weeding out the misfits. It could be the math itself works it out. It could
Right.
be the shape of the spiral can't be anything other than what it is, because that's how that spiral grows. Just because it can't grow in any other way. But why is that any less magical or miraculous or divine? So
Yeah, it could very well be that Plato was right, and there are these set of forms that create all these different things. So that he talked about the head of the horse and how it just over time just took the same basic shape and morphed it around till you have the horse of today from animals, hundreds of thousand years ago. Um,
I think they started out about the size of a cat.
yeah, they were much smaller. I don't know how small. So the next guy we're going to go to is a chap at Yale, Harold Burr, and he apparently was the first one who got some traction with realizing that electricity had some influence on biology. Um, so he actually published a paper on an electrodynamic theory of development, which is the one that's widely quoted. And this led him to have an electrodynamic theory of life, not simply development, and electrical characteristics of living systems. And he used a voltmeter, but the voltmeter that he used uh, detected the electrical potential of the body, which he reported. Other people, I'm sure Tesla had done this, okay, but the the end result of this was that Burr came up with this term of an L field for biological electric fields in living systems. And every cell that you have in your body has a slight, it's 20 to 50 millivolts potential on it. So you have 50 trillion cells, so there's no shortage of electricity floating around in your body, but it just gets ignored. Is like why do you have this? Doesn't it bother you that you don't know why? But Burr really went at it, and in 1942, uh, he looked at corn, and he noted that electricity seems to bridge the gap between the lifeless world and the living matter world, and it's really a key factor in all living systems here. And this led to the electrical detection of cancer cells, experimental embryology, neuroanatomy, and the regeneration and development of the nervous system. He was trying to fix nervous disorders, regenerate nerves. His studies of bioelectrics of ovulation and menstruation eventually led to the marketing of fertility indicating devices, and his late studies of the electrodynamics of trees, carried out over decades, suggested entrainment. The trees are tracking diurnal, lunar, and annual cycles, and so forth. So let's see. I have this thing here. Let's see if I can get this up. Hold on.
Makes perfect sense. And
The paper
Including Burr and Northrup's The Electrodynamic Theory of Life, 1935, by Michael Levin, published in the journal Biological Theory in 2020, offers a
comprehensive review and modern interpretation of Harold Saxton Burr's groundbreaking work on bioelectricity and its fundamental role in biological organization and morphogenesis. Levin highlights Burr's visionary perspective, which posited that electric fields are not merely byproducts of cellular processes but active determinants of life's organizational patterns. This reevaluation
Bruce, can you hear this?
Yeah.
Okay.
places Burr's work within the context of recent advances in the field, demonstrating its prescience and continued relevance. Key insights from the paper: Burr's pioneering contributions. Burr is recognized as a foundational figure in developmental bioelectricity, exploring how organisms from trees to human beings are deeply influenced by bioelectric fields. His research spanned several decades, from the 1930s to the 1950s, and laid the groundwork for understanding how electric fields guide growth, development, and healing. Bioelectricity as a guiding force. Levin elaborates on Burr's hypothesis that bioelectric gradients serve as pre-patterns guiding morphogenesis. This idea, initially speculative due to the technological limitations of Burr's time, has since been validated by contemporary molecular physiology, affirming Burr's vision of bioelectricity as a crucial player in biological organization. Implications for health and disease. The paper discusses Burr's insights into the relationship between bioelectricity, health, and pathology, including cancer. Burr's observation that bioelectric patterns are altered in the presence of tumors presaged current research into how bioelectric signaling can be leveraged for diagnostic and therapeutic purposes. Philosophical and methodological contributions. Beyond his empirical contributions, Burr is credited with integrating concepts from physics and philosophy into biology, advocating for a holistic approach to understanding life. His emphasis on the importance of electric fields anticipated current interest in systems biology and the study of non-local interactions in organisms. Legacy and future directions. Levin suggests that Burr's work offers a rich vein of ideas yet to be fully explored, particularly regarding the use of bioelectricity in regenerative medicine and synthetic bioengineering. The paper calls for continued research inspired by Burr's interdisciplinary approach to uncover new mechanisms of bioelectric control in development, regeneration, and the treatment of diseases. In the shadow of modern science's focus on genetic and biochemical narratives, Harold Saxton Burr's The Electrodynamic Theory of Life, co-authored with FSC Northrop in 1935, emerges as a pioneering exploration that bridges the gap between biology and physics. This theory posits that life must protect its bioelectric fields to sustain itself. Given the potential for artificial EMFs to interfere with the natural bioelectric fields of organisms, it becomes important to consider measures to protect these intrinsic fields. This perspective aligns with Burr's insights into the significance of bioelectricity in life's processes and underscores the importance of environmental health in the broader context of biological well-being. Practical steps can be taken to minimize unnecessary exposure to artificial EMFs. Learn to protect your bioelectric fields from sources of EMF like cell phone radiation at rfsafe.com. The paper, Revisiting.
So there you have it. Burr launched this activity, and let's just go back here and see. Yeah, here it is. Oh wait, it's here. That's where it is at. Okay. What happened here? Let's see. Can you see this, Bruce?
Yep.
Okay. Burr got really caught up with this, and in 1962 he wrote a book about the nature of man and the meaning of existence, which was an attack against materialist philosophy. So he said there's an order in the universe, there's unity in the organism, and man is endowed with a soul. And his book, The Blueprint for Immortality, which was published late in his career, though he based it upon work carried out over decades, contended the electrodynamic fields of all living things, which may be measured and mapped with standard voltmeters, mold and control each organism's development, health, and mood. And these are what he called living fields, or L fields. And he compared the L field to what this other chap, Hans Striesch, calls the morphogenetic field. And Burr used this L field to explain cellular differentiation and the form of living organisms. So his theory has been rejected by most scientists. But most things are wrong, but they suggest a new direction to go in, and he's probably onto something here, and so forth. The other thing is he did this. You can see down here, he's got this box here. When you look at modern electronics, this box is garbage. It's amazing he could measure anything whatsoever. Just putting the electrodes onto these different entities and not having the measurement process itself really distort things. It's amazing that he is that good an experimentalist that he was able to overcome all of these problems. But he's a very key figure that we'll come back to. Then there's another Brit, Dennis Noble, who's he's actually at Oxford from 84 to 2004, and he's a professor emeritus of computational physiology and a pioneer of systems biology, and developed the first viable, why is this thing going? There we go. First viable model of the working heart in 1960. And so he's been attacked by a lot of people for being a nut. So let's just take a look at what.
Think of how life must have emerged.
Hold on a second. I just want to make sure we get this thing. Here it is. And we got sound. Okay. Okay, here we go.
Long ago in, we don't know exactly where, perhaps in the minute fissures and cracks in thermal vents, who knows? But until the living system, the networks of interactions that were forming autocatalytic sets, meaning chemical reactions that could continue indefinitely, until they were enclosed within membranes, they would have just dispersed. So somehow or another, that constraint, the forming of a membrane around a catalytic set, would have been a key element in any form of evolution of what we call life. Think of how life must have emerged long ago in, we don't know exactly where, perhaps in the minute fissures and cracks in thermal vents,
So
who knows?
This is repeating, okay? So what he's saying here is that initially there was just this chemical soup, and somehow this membrane formed around it in different places, and that created what today we call bacteria. Bacteria are single-celled creatures, but we're multi-celled creatures. So the next thing happened was somehow the single-celled creatures butted up against one another and merged, and the two different cells took on different functions inside one another, and that started to create a lot more diversity, where each of these cells would focus on one thing. Like, I'm going to be a muscle cell, I'm going to be a neuron cell, I'm going to be a skin cell. And that really leads to where things take off. So you get a taste there of how Dennis Noble ends up thinking very, very deeply about this stuff, and going back to really fundamental questions. And of course, this doesn't make him popular with these other people. So let's see, what's the next one? Oh, then we come to Michael Levin. He's at Tufts University now, He actually was born in Moscow, and his parents were Jewish. His father was a computer programmer, and they just because of the attitude that the Soviets had toward Jews, they immigrated first to Israel, and then he actually lives up on the north coast of Massachusetts in Marblehead, Massachusetts, which is a very scenic place. And he was at Harvard, and he got poached on to go to Tufts, which is just a few miles away from Harvard. And so his research interests include bioelectric signals, building on this work of Burr and others. And the cells communicate with these bioelectrical signals to handle their anatomical needs during development, during regeneration, and during cancer suppression. You know, every day you have probably 10,000 different cancer cells in you, and your body wipes them out. You know, in fact, it's a miracle that any of them get through. And obviously it does. It's one of the leading causes of death due to disease. But it just shows you how fundamentalist can be. The cognition and intelligence in diverse and unconventional substrates, okay? That there could be in actually the dirt that you see around you, there might be cognition and intelligence. And then a much more top-down control of form and function that has to scale across all the different scales in biology, from cells on up to, you know, major animals, elephants and whales and so forth. Paul Allen, who is one of the co-founders of Microsoft, gave him a $10 million fund. So he said, I don't want you to waste time writing research proposals. Here's this money. You just go work on what you want to work on. And so that's why he has this Allen Discovery Center at Tufts. And the other thing that Michael Levin is known for is he discovered what he called xenobots. So these are living robots that are made from frog skin cells that can sense their environment. Okay, so they're literally a hybrid. You know, you could say it's a living robot, but it's made out of frog skin cells. Okay, it's not made out of metal or anything else. There's a xenopus, which is an embryo culture system that should enable the discovery of new drug targets and the development of therapeutics. And then when you combine this with what he calls multiomics and an integrated bioinformatic that is a combination of computer technology and biology, then you end up with some interesting things happen. So as of 2021, which is just the last time he updated his website, his lab with all of his students is working on synthetic biology applications of bioelectricity for controlling the cell, the development of bioinformatics of shape, which is D'Arcy Wentworth Thompson, and being able to morph all of these different shapes around in different ways, using AI for discovery and testing of algorithmic models, linking molecular genetic data to what's called morphogenesis, and then again using techniques from AI and computational neuroscience and cognitive science to make models of morphogenesis. I just saw an article on how AI was picking up a lot of the traits of intelligence, and I just sent it to him 2 days ago, and he replied and said thanks. You know, he's that sort of guy. He's eminently accessible, but he thinks very, very deeply about this, and he has attracted a lot of really high-powered students. So I fully expect he'll get the Nobel Prize in biology someday. The only question is when.
I just hope these ideas aren't used for evil.
Oh, they will be
Yeah,
the
seems
the bad
impossible.
guys that are out there, you know. Yeah, I mean, we already saw.
You're gonna
They-
create slave cells
Yeah.
to do God knows what.
Right, I agree. So now we turn to Robert Becker, who wrote a book that I read a number of years ago called The Body Electric, Electromagnetism and the Foundation of Life. Now you can buy this book, but in fact, it's available on the web for free as an Acrobat file. So you don't need to pay anything for it as long as you want to read it as an Acrobat file. And he after World War II, he was a professor at the State University of New York, or SUNY, the Syracuse campus, and is the director of orthopedic surgery at a veterans hospital that was there in Syracuse. And this led him to look at how to regenerate. He's dealing with a lot of people that had, because of the veterans hospital, a lot of injuries. And so maybe he started playing around, seeing, is electricity possible to end up helping to regenerate things? And he carried out, his book describes an amazing research program he carried out, but it came to nothing because he couldn't get funding for it anymore. He he was very active in opposing high voltage power lines because he felt that if you really believe that this bioelectricity is key to shaping your health and your body, then these high voltage power lines are going to really mess up your body. And there's a lot of evidence that that is indeed the case. Or cellular phones going from first generation to second generation. Now we're up to fifth generation, going to sixth generation. The power levels keep going up with every level, and you're putting this right next to your head, and it's the damage is undoubtedly incredible in terms of it's torquing all of your DNA that you've got whenever there's any division of cells, and nothing good is going to come out of this. So Becker is an interesting guy. It just is an example of what he came up with. He actually measured electrically all the meridians on your body. There actually are resistive paths that connect all these different parts in your body, and so he actually was able to map all the meridians that are used by acupuncturists. But the measurement itself is incredible. So it's not something that a unskilled person could carry out to actually see where is the meridian going. And it's a different thing. What he did was he put some headphones on someone who was laying down, and he discovered there was a particular set of frequencies, and if you played these, the person immediately went to sleep. And when you quit playing it, they woke up. So here you had a way to do, for example, in surgery, there's always this issue of giving people some sort of anesthetic to make them relax so that they won't move during the operation. Well, if you could do this without any drugs by just playing these tones, that might be of interest. But if that's been attacked by a lot of people, but there probably was something there. People have had trouble duplicating some of the things he's got in this book. And another side to it is he spent a lot of time with salamanders and how they ended up having pieces of their body cut off, and he carried out electrical measurements to see what was going on. So in this particular book here, the first part discusses regeneration in salamanders and frogs. And he did things where he would amputate a limb, and then he hypothesized that the electric fields were playing a role in controlling the regeneration. And he mapped out what the electric potentials were during this regeneration and so forth. And when the limb of a salamander frog was amputated, he measured what the voltages were which came to be called something. It went from minus 10 millivolts to plus 20 millivolts or more, which is a complete flip of 30 millivolts. So this was called a tip-off that there was an injury. And in the frog, the voltages simply changed to the normal negative level in 4 weeks or so, but there was no limb regeneration. But in the salamander, the voltage during the first 2 weeks would change from plus 20 to minus 30, and then go back to minus 10, and the limb would be completely regenerated. So there's a tip-off that something is going on here, but it's not clear what. We don't know enough about the mechanisms for doing this. But again, all of these mechanisms have to be in the DNA. So how is the DNA electrically interacting to create these things? Becker found that regeneration could be improved by applying electricity at the wound, and that the bone had some piezoelectric properties. That is, when you put an electric field on it, it mechanically torqued. And so this would generate a force that would generate a healing current, which would stimulate growth at the stress locations here. That was one thing he came up with. He described the potentials in the magnetic fields in the nervous system, taking into account the Earth's magnetism and solar winds. And he also measured along the skin, like we said, for the meridians and acupuncture. And then at the very end, he went through why he's very much against electromagnetic, high intensity electromagnetic fields, or what he calls pollution. And so it goes. So that takes us back to Michael Levin. This is just some of the things that he talks about here. Each of us took the remarkable journey from matter to mind. Once we were a quiescent egg cell, slowly we became human beings capable of advanced metacognition. And so his group studies these processes by which intelligence in myriad conventional and unconventional embodiments operates in the physical world. And they combine developmental biophysics, computer science, behavioral science to understand how cognition scales up from the metabolic and physiological competencies of single cells through the organ building and repair capabilities of cellular collectives to the classic behavioral repertoires of whole organisms and swarms. So they're always looking for scale invariant principles, things that are true at all levels, from single cells on up to huge herds of elephants. And this involves a lot of cross discipline activity. The main model is what has been called morphogenesis, the ability of these multicellular bodies to self-assemble, repair, and improvise novel solutions to anatomical goals. And so they're dealing with mechanisms required to achieve robust, multi-scale adaptive order while they're alive. And what are the algorithms that are sufficient to reproduce this capacity? One of their unique specialties is including this bioelectricity, the ways in which all cells connect, their electrical networks that store, process, and act on information to control the large-scale body structure. And finally, they create tools to read and edit this biological code that you could think of this as the software. The body is the hardware, but this is the software that is actually controlling it. And again, they're there at the intersection of all of these different activities. They're looking for general principles of life as it can be, and so forth. So one of the things I liked here was there's spatial information and temporal information, and these need to be detected, remembered, processed, etc. And so they carry out a lot of tools to allow them to do this, etc. The thing that we talked about, morphogenesis, he came up with a block diagram of different entities that are interacting with one another and trying to relate this system here as in fact agents. So over on this side is how do they know what to do? That's all these things. These things, how they know what to do. And then how do they do it? Well, they do it this way with these things over here. So, and so it goes. And there's a huge amount of activity that he manages in this. And it's amazing that he has an incredibly active life as well, doing all kinds of things. So, he created these xenobots, which we'll come back to, and then beyond growth and form, the thing is that all intelligences are collective intelligences. That is, they're made up of parts, and therefore life uses a multi-scale architecture. Each layer does something different to solve problems here. And William James said the ability to reach the same ends by different means. So that's what they're looking for, is how the body does these different things in different ways. And so it goes. So I just got some, let's just take one of these. There's a bunch of short ones. And I think what we'll do next time is do some of the longer ones. But
something we discovered years ago that...
Bruce, can you hear this?
I can hear it.
You can, okay.
Tadpoles need to become frogs, and in order to do that, they have to rearrange their face. So they have to move their eyes, their mouth, their nostrils, everything has to move around. And it used to be thought that this was a hardwired process. Every one of these organs moved in the right direction, the right amount, and you get your frog. So this is why it's important to do experiments in these things and not just assume. What we did was we created so-called Picasso tadpoles, where we basically scrambled all the craniofacial organs. So here the eyes on top of the head, the mouth is off to the side. It's totally scrambled. Well, these things, it turns out, remodel into quite normal frogs, because all of the organs will move in new paths. In fact, sometimes they go too far and actually have to double back until they get to a correct frog face. Something we discovered years ago, that tadpoles need to become...
So that's just one example there. Let me try the next one here. Here we go.
What's one of the biggest myths in biology that you, with your research, with your...
Hold on a second. I forgot to do screen share. Okay, can you see this, Bruce?
Yep. Okay.
Let this soft mineral melt in your mouth to rebuild your gums and teeth, and never need a dentist again.
What's one of the biggest myths in biology that you, with your research, with your lab, is helping overturn?
Yeah. Hey, Kurt. Great to see you again. I think that one of the biggest myths in biology is that the best explanations come at the level of molecules. So this biochemistry, what they call molecular mechanism, is usually currently taken to be the kind of the gold standard of what we're looking for in terms of explanations. And I think in some cases that's valuable. That's that has the most value. But in most cases, I think it's not the right level. And Dennis Noble has a really interesting set of papers and talks on biological relativity. So this idea that different levels of explanation, especially in biology, provide the most bang for the buck. And I think in terms of looking forward as to what do these explanations let you do as a next step, I think we really need to go beyond the molecular level for many of these things.
What's one of the...
So there you go. The mainstream view is very much, again, driven by molecules, by biochemistry. And one of the problems that you encounter in this is the complexity is off the charts in terms of doing all these things. But, there's so much money available for this that what can you say? So let's try another one here. What is bioelectricity?
Cells have the ability to generate a resting potential using ion channels and pumps in the membrane. And they have these little electrical synapses known as gap junctions, which allow them to share their state with their neighbors. Mm-hmm. And both the channels and the gap junctions can themselves be voltage sensitive. So now what you have is a voltage gated ion conductance, which is basically a transistor. That gives you a kind of historicity. It gives you feedback loops, memory, right there, right there. And when these cells connect with each other into electrical networks, you get some of the same phenomena we see in the brain, just on a slower time scale. So these are not millisecond, these are hours, on the scale of hours. But there's all kinds of amazing information processing that these networks can do that allow them to pursue. Cells have the ability to.
So that's another short one. Okay. Let's just try.
And just to point out, if anybody's interested in plants, this is another great example of the plasticity of life. You might think that the oak genome encodes for this shape, because this is what you see most of the time. These acorns give rise to exactly this shape, this flat green thing. But along comes this non-human bioengineer, this wasp, which is a parasite, which puts down some signals for the plant cells, and it hacks them. It hacks the morphogenesis exactly the way that I've been showing you that we can hack the morphogenetic outcomes in animals, and it causes these plant cells to build this incredible spiky round thing, or even this stuff. So these are galls, and we would have had no idea what these cells are capable of if we didn't see this amazing example of bioprompting or reprogrammability. And in fact, it's kind of interesting that the sophistication of what is built is roughly parallels to the sophistication of the hacker. So the last example I want to show you is a new kind of technology. And just to point out, if anybody's interested in.
Okay.
This is another great example of the plasticity of life. You might think that the oak genome encodes for this shape, because this is what you see most of the time. These acorns give rise to exactly this shape. It's a flat green thing. But
okay, sorry. So you start to get a flavor for what is Michael Levin doing, and this is definitely not this mainstream stuff that we were talking about. And so it goes. There's a bunch more here, but I think we'll just hold off for now. You get to see what's going on, and that there's clearly something here, but we don't know what it is yet. But probably in another 10, 20 years, it'll come into focus, and who knows what we'll have. We'll have medical beds that you lay down in the bed and it just heals everything, and you become 20 years old. Who knows? So this is to be continued.
But he's in the, let it be noted, he's in the D'Arcy Wentworth Thompson lineage and not the Darwin.
Well, he's aware of all what Darwin did, and he's aware.
Right.
He sees all this other, biochemistry, and he looks at all the genetic stuff. I mean, they're developing tools that are incredible. If you put billions of dollars into something, sometimes get some good stuff out. But he's not. This is what always amazes me about these Russians is what school do they go to that they learn how to attack problems this way? There isn't any school I could think of that. Where would you ever, come across doing things this way? So
maybe it's in their genes.
Yep. So have a wonderful time, Bruce, at your retreat, and we will adjourn for a week and watch your email when you come back, and you'll see part 2 of this, where we go through some more.
All right.
You can see a lot of good stuff here. Do
you see why people want to stop?
No, we don't want to. Well, I mean, Marjorie brought it up. I realized we hadn't done justice to this whole area. Okay, and this is real medicine.
Yeah, no kidding.
This is if you want to live forever, this is what you should dig into to see. So
Well, I feel it's our whole thesis in a nutshell of what we've been working on for the past 5 years. What we've been really wanting to share is the sense of wonder.
An order. Look at the order that's there.
Right.
At every level. It's just unbelievable. So someone, this is a Picasso to end all Picassos that created this. So, okay, have a good. We'll see.
All right.
Bye-bye.
Bye. Safe travels.
Thank you.